Good afternoon. I'm Dorothy Gray. Welcome to this news briefing from the 254th National Meeting and Exposition of the American Chemical Society in Washington, D.C. We're joined today by Dr. Mark Blenner from Clemson University. He's studying ways to recycle astronauts' urine and exhaled carbon dioxide to make chemicals needed on board for long-distance space flight. Dr. Blenner?
Thank you for asking me to be here today, and I'm excited to get to tell you a little bit about our work that we'll be presenting tomorrow in the Journey to Mars Symposium. So my group is involved in what's known as synthetic biology, and we are trying to basically engineer microorganisms to make valuable molecules from low-cost feedstocks. And we got interested in how we can use this sort of biotechnology to help astronauts go farther away from Earth and spend more time in space. And so if we look at how astronauts are surviving in space right now, they're heavily dependent on Earth for their survival. Things that are shipped from Earth that they use to generate their oxygen, that they use to eat, et cetera. And so as we start to send astronauts farther away from Earth for much longer periods of time, they're going to have to learn to be independent of Earth. And that's where we think our biotechnology and similar biotechnologies can play a role. And so microorganisms have an amazing capability of diverse chemistries and can produce all sorts of different types of molecules that are going to be critical for astronaut survival. And so we looked at that potential and said, great, but then what's the challenge here for us? We know we can do a lot of this kind of synthetic biology on Earth now. So the tricky part is, how do we do this in space? And the critical need here is the need to provide the nutrients to grow microorganisms that are engineered to make a variety of different products. And so when we thought about what was available in space, we realized that there'll be resources wherever we go. So if it's to Mars, we think there'll be some carbon dioxide there. But in transit, there's really not going to be a lot there. It's going to be whatever the astronauts bring with them and basically the waste they produce. And so we started thinking about how to take the waste products produced by astronauts and find ways of putting them back into a form that microorganisms can then use to grow and to produce the molecules that astronauts need again. And so we've really taken a look at two examples of wastes, the first waste being human urine. And the reason we looked at that is it's relatively abundant. It's a relatively abundant waste by the astronauts, and it's rich in nitrogenous compounds such as urea. It's actually the major component other than water in urine. And that nitrogen is an important component of microbial cell growth and production of valuable products. The other thing that microorganisms need to grow really is some source of carbon. And so we happen to work with a type of yeast. It's a yeast known as Uroea lipolytica, and it unfortunately does not grow on carbon dioxide directly. And so we've sort of thought about how to get carbon dioxide, that is an astronaut waste from their breathing, as well as a potential resource that could be obtained from the Martian atmosphere. How do we get that into a form that our yeast can then use to grow? And there's a couple of different ways of doing that. Obviously, things like plants would fix the carbon into more complex molecules like carbohydrates that then could be used to grow our microorganisms. We chose cyanobacteria and algae as an example of a way of taking the carbon dioxide out of the atmosphere and putting it into more complex carbohydrates and lipids that our yeast are good at growing on. So right now we've actually done some really interesting work with the human urine, showing that our yeast not only can grow on the human urine, they're not inhibited by a lot of the compounds that are in the human urine. We knew at the start of this study that they would grow on urine. We didn't know how well they would, and it turns out that they prefer it actually over other common nitrogen sources. And so that was an interesting discovery that we've made. We're still sort of working out a lot of the details on how to take advantage of the cyanobacteria carbon fixation process. So we can get cyanobacteria obviously to grow on carbon dioxide, and we've been able to use it to replace certain components of microbial media for our yeast, but there's still a lot more value and a lot more of the carbohydrates that are locked up in the cyanobacteria mass that we haven't yet been able to access. But we're confident with some more work we're going to get there soon.
Ready for questions?
Yeah, and I guess that's my spiel. So if there's questions, I'm happy to field questions right now.
We'll open it up to questions here. Please state your name and affiliation before asking your question, please.
Hi, so it's Kath O'Driscoll from Chemistry and Industry magazine. When you said about the nitrogen content of urine, it immediately put me in mind of fertilizers. Is it going to be possible to actually make fertilizers from this type of thing? How do you envisage that might be possible? So astronauts, I'm thinking, could then grow their own plants and food.
Yeah, so there's a lot of ideas right now about using human urine for fertilizer. I think if you're going to turn it into a fertilizer, I do believe I don't really know the plant area so well. From what I understand, that there's going to be some capability of plants using urea as a nitrogen source. Just really, I think a lot of it has to do with the community of microorganisms that are in the soil. We haven't really been thinking too much about how our bioprocess would sort of how that really interplays with the production of plants and crops. But there's certainly going to be links there that we're just not ready to explore ourselves. But I do think that there's potential for either, using the urine or or perhaps some of the byproducts of our process in fertilizer applications.
And the press release also mentions that you're looking at making polyesters. Can you talk a little bit about that?
Yeah, sure.
How many different types of polyester and why polyester?
Yeah, I realized I hadn't talked anything about the products that we're producing. I just babbled on about the urea and the carbon. So the products that we're trying to produce, again, these are just two examples of products that we think there'll be some potential for need for astronauts. So one of those products is omega-3 fatty acids, and omega-3 fatty acids are important for astronauts in its implications in prevention of bone loss. It's also been implicated in cardiovascular health and ocular health. So there's a number of reasons why we're interested in this kind of a compound. And there's just really no other source for it. It's also somewhat unstable, and we're not really sure how long it's going to last in long-term space flight. There's it was the example we came up with because we had already been working on a process to make omega-3 fatty acids for terrestrial-based applications. But there's a number of other vitamins that have relatively short lifetimes, such that shipping them to Mars or shipping them deeper into space is actually not going to be possible, just based on the amount of time in transit. And so there has to be some sort of a process to make those kinds of compounds. And Whole Foods is definitely one of those ways. And we think there's potential also for microbial production of those kinds of compounds. The second thing that we're focusing on making are these polyester plastics, and it was kind of a simple first demonstration that we could take these astronaut wastes, convert them into something useful for microorganisms to grow on, and then we've engineered the same yeast to produce the polyesters known as polyhydroxyalkanoates. So right now, the types, you're asking about the types of polyesters they make. The properties of these polyesters depends on the composition of the monomers that go into the PHAs or polyhydroxyalkanoates. These are actually intermediates of fatty acid metabolism, and so they basically take on the composition. They take on a composition that's a function of a number of different parameters, including the fatty acid composition that the cell likes to maintain, as well as the specificity of, there's an enzyme we've introduced into these yeast to polymerize these intermediates. And those enzymes also have some amount of specificity. So right now, the polyesters we're making, they have carbon chain lengths of on the range from C10 to C14. We haven't been able to harvest, and we haven't tried yet. We just haven't harvested enough material to do any kind of testing. So we don't really know what the properties of this material are yet.
All right, so they're novel polyesters then, are they? Because I was thinking, would they be polyesters for sort of repair applications if something went wrong on the craft, or if they needed surgery or something like that?
Exactly. I think there's a number of different applications for it. Our original idea was that these kinds of polyesters might have some application in 3D printing. And so if an astronaut needed a tool or a part, they might be able to fabricate it basically from their waste. But they're not exactly novel. I don't think it's fair to say they're novel at this point, but they're not largely abundant, really. And so we haven't really been able to, not just us, I think in the field, we really haven't been able to produce much to do any kind of significant testing on. So the properties of it are still a little bit, I'd say, underexplored. And having the biological organisms, the ability to engineer these biological organisms, we are interested in exploring, I guess, different genetic manipulations that will allow us to change the monomer composition so that we can try and tune the properties once we figure out kind of what our baseline is and where we need to go.
Okay, thank you.
In the back of the room, please. Thank you.
Ben Valsler, Chemistry World magazine. I was just wondering if you could describe the reaction vessel you're using to do these experiments and what you think it would take to make that something that could go on board, the International Space Station or whatever vessel is going to Mars.
So right now in we're doing this at very small scale in our lab, and so the vessels are simple shake flasks that are no larger than this cup. They're 250 ml shake flasks typically. We try not to go up in scale unless we really have a reason to, and right now we're still demonstrating the technology, and so we haven't had a need to start to move up in scale. The Cyanobacteria Reactor is basically, it's a bottle that has some air purged into it and spins, and it's got air purged into it. It's also got a spinner. So these are very simple looking vessels. They would not probably be appropriate for a microgravity environment. So unless you wanted all your microbial cultures everywhere, we would need more sealed vessels. I think there'd have to be some interconnection between the yeast vessels which produce CO2, the cyanobacteria vessels which consume CO2. Oxygen might need to be exchanged between them. It gets really complicated really quickly. To implement anything in space requires a real high level of sophistication and sort of process integration and making sure that your systems are reliable, simple. We're not there yet. I think it's going to take a lot more work just for the vessels alone, let alone I think it's a little bit unclear with how the biology is going to react to being in space. Mm-hmm. That's an open question right now as well.
What sorts of yield are you getting? So when you've put in a portion of human urine and you're getting out your product, is it likely to be enough to sustain a spaceflight crew?
It depends on what we're talking about in terms of products. I mean, the short answer for everything right now is no, but that's again, I think it's going to take some of our effort and time put into trying to really redirect where the carbon flows inside of these cells. It's I think completely feasible for the omega-3 application. It's sort of already been done on Earth. So the needs for things like nutrients are much lower than, for example, the needs for fabricating a part. I think I had been asked a question before about how much, at our current yields, how much of the yeast would it take to produce like a small wrench? And I did the back of the envelope calculation, and it would be 1,000 liters. And so clearly it's not going to work in space right now, but we do have to, I am encouraged at the starting point, and we have a lot of reasons why we think that we can get to much higher yields, and by getting to those much higher yields, then we're in territory where it would actually be feasible. The type of yeast that we work with is really good at storing and accumulating lots of fatty acids and lipids and intermediates of fatty acid metabolism. There's been studies showing that they've been engineered to accumulate greater than 90% of their weight as lipids. And so if we can get even close to that with accumulation of plastics, which are actually just intermediates of that same metabolism, then we'll probably be in pretty good shape. But it's going to take a significant effort, not to downplay how hard it's going to be, but I do think it's possible.
Okay, thank you.
Yep.
Next question, in the front row.
Kathy Kowalski, freelance reporting for Science News for Students. Could you backtrack a little bit and kind of explain these yeast make the fatty acids and the polymers, the polyester compounds, are intermediates of it? So are these yeast just naturally little fatty acid factories, and have they already been used to spit things out like E. coli does now, or is that the novel direction that your lab has to go in?
It's a really good question. So the yeast have a very, I would say, it's probably pretty accurate to describe them as little fatty acid machines. They're basically, what they're really good at is they will store excess food when they don't have all of the nutrients to grow. They recognize that they have excess carbon in their environment and will take that up and store it as fat. I like to think that we're kind of similar in that way. So basically these yeast have metabolism, they have biochemistry, which makes them really good starting points for producing any of these types of molecules which are derived from fatty acids. The polymers here, the plastics, are, if you look at how these organisms degrade fatty acids, how they basically eat the fatty acids when they are able to, they use a cycle known as beta oxidation, and one of the intermediates of that process, which is actually really efficient in these yeast, produces this compound, these hydroxy acids, which are the monomers that then get polymerized into these polyesters. And so we're basically able to sort of naturally, these yeast are pretty good at pushing carbon into lipids and fats, and then are also very good at taking those and breaking them back down for energy, and for building blocks to build more cell material. So we just kind of hijack that process and tweak it and make it focus more on doing that rather than growing at that point. And we do that through a number of different genetic engineering tools that my group and others have developed.
Okay, so it's little yeast makes the fatty acid. You want to get it to break it down right away. That gives you lots of building blocks for your polyester polymer. Mm-hmm. And then you got to make the yeast give them up to you.
Yeah, so this is a great, okay, so you're addressing the question, how do you get the plastics back out? And that is right now, we know how to do it, it's just not really a space-ready application. It's a solvent extraction to get them back out, but that's not really attractive for simple, reliable
you want to keep these
processes.
Guys alive as you're zooming through space.
Right. Well, I mean, you do and you don't. You don't want them, you can definitely live with them dying. You can harvest the cell mass as long as they reproduce and you still have some left, right? If you have some left, they'll continue to grow if you give them the right nutrients and make more cells. And that's actually one of the most attractive aspects of using biotechnology for sort of manufacturing in space, is that to start with, you don't really need a lot as long as you have a source of media to grow them on. And so it ends up being if you can make your media on site, it ends up being a relatively low investment of mass and space. But you're absolutely right in that it's not going to be easy for us to figure out how to get the plastics out of the cells. Definitely not if we want to keep them alive. I know there are lots of people interested in finding ways to get these cells to give up or spit out their fats and fatty acids and other products they're making. They're not really good at that. They're using these normally as storage for a later day, so they're not going to die in the process normally. So we have to kill them basically to get out the plastics. A kind of more far out there idea we've had is that if you can really push the accumulation of those kinds of polyesters to a much higher percentage, where the cell is maybe 70-80% polymer by weight, you may not have to get them out of the cells, maybe, and this is not a question I'm ready to address, but it's possible in my mind that you could actually print the plastics as part of the cell, but that's really just a way out there concept. Yeah.
Bela Buslik, ACS Communication. These ideas sound really fantastic to recycle stuff in the space station and all that thing. But then you've got to do something with it. The raw materials, of course, come from the people that are populating the space station and so forth. But they have to be resupplied all the time. Otherwise, there's nothing to eat. I'm sure. I'm not sure, but you may have heard Biosphere 2. That was a grand experiment. Led to the point where when the CO2 levels were roughly about the 4 or 5% level, and the oxygen down to 16%, they had to quit the experiment because people started getting sick and dying, because there's just not enough space up there. And with Biosphere 2, the major problem was they represented the continents and everything, just not enough water. And so how do you get around something like that when you're trying to go from here to Mars?
Another really good question. You guys asked lots of great questions.
I know it's an unfair question because each individual piece is marvelous. I mean, that's a great way of recycling thing. But what do you do with all the plastic once you've got other than bringing it back to Earth?
Yeah. So right now, I mean, no one's really designing materials for use in space with any recycling in mind. And so part of the case that people like me and people who are interested in what I'm interested in are trying to make is that, as we now have we're exploring options really for the future. It's not clear that any of my technology will ever be used. It's just one of many possible solutions. But while we're doing this exercise, it makes a lot of sense for us to start thinking about making materials which are easily converted back into forms which can then be either reprinted or actually fed back to the microorganisms. And this is probably one of the benefits of using these polyesters I'm talking about, which are they're biopolymers. They're easily biodegradable. That of course has its own challenges as well in terms of how easy it would be probably for them to be broken down by the microorganisms. But this is really an opportunity to sort of rethink what we're doing already and try and integrate more of this sort of closed loop thought process into basically anything that's going to be brought that far into space. To some degree, I mean, you're never really going to be able to close these material balances correctly, and so there's going to be some need for additional material, additional atoms coming into your system. A lot of the thought right now is that we can harvest that kind of stuff from Mars to some degree. Particularly the CO2, there's a good bit of, there's not a huge atmosphere, but it's mostly CO2. There's a lot of assumptions going into things right now about the availability of water on Mars that could be split. I think it's not necessarily well established that we're going to have that capability or that there's really that much water to be had, but you kind of, again, start somewhere. So there's no easy answer to your question. I think if I had the answer, we would be going much quicker to Mars. But,
I think we have another question over here.
It was just a quick question. I think you already sort of alluded to the fact that you still don't know what might happen to the biology in space. I was going to ask, have you done any work with these systems, with the yeast in microgravity environments? And also, is it known whether a 3D printer, because I know fluids behave very differently in microgravity, so is it known even whether the 3D printer would work?
I think so. The first part of the question is there's been very little. We have not tested our own yeast in a microgravity environment. I think there have been very few organisms that have really been studied at all in a microgravity environment, and the ones that have, they tend not to be, I don't know this for sure, but my reading of this was that there weren't very many genetically engineered organisms going up into microgravity studies, and so it's not entirely clear either if how stable these genetic engineering modifications are going to be in the space environment. So there's definitely a lot of just basic space biology questions that need to get answered before this really gets to be more realistic. I'm sorry, what was the second part of that
About
question?
whether the 3D printer would
Oh.
work, and I'm thinking really because of the different behavior of liquids. And have you any plans to do any microgravity?
There's a company called Made in Space that I think has developed and has tested already on the International Space Station a 3D printer that addresses that exact problem. They've not tested our types of polymers in it, but I think that NASA is making significant investments in trying to get additive manufacturing working in space and in microgravity environments. We honestly do have plans to try and do some testing of our organisms in microgravity. That's all dependent on funding and opportunities, but we would definitely like to see our system get put up in space.
Thanks.
Thank you. The archived version of this session will soon be posted at bit.ly/ACSLive_DC. Please join us for our next press conference at 9:30 a.m. Wednesday on eradicating mosquitoes by luring them with sweets. Thank you.